To see everybody saying hi. I said my name is Jade Edelstein. I'm marketing manager at GeneYX, and you'll see that Dr. Hagar Moshaked and Dr. Ali Tabish is also with us. Hi, Christina from Spain. Welcome. Okay, so it's going to be a busy course. I'm going to start. It's now 6:30, and we've got a lot to get through. So just give me a second. And then I think we should go ahead. But I do see that more people are coming in. I'm going to start with an introduction, just, some housekeeping and how things are going to move for this course. So people will not be missing out on any, important course material. Okay, very exciting. I'm going to start in 20 seconds. Welcome everybody. We have 193 participants with us this evening. Very exciting. And welcome to everybody from all over the globe. This is the first lesson of 9 on the Clinical Interpretation Analysis course. And I wanted to start by letting you know that all lectures will be recorded. And I will send you through a copy of the lecture at the end of every week. These recordings will also be used for training and marketing purposes. I would like to firstly introduce myself. My name is Jade Edelstein, and I've spoken to many of you via email and LinkedIn. I'm the marketing manager for GeneYX, with our CMO, Raviv Yitzhaki, who is also in our audience. Because we have many participants, I have muted everybody, but please feel free to use the chat feature when and as you need during the course. So here's a little bit more about GeneYX and what we do. GeneYX was founded in 2018 and is used in 36 countries. Our team has analyzed over 300,000 samples, building valuable insights for hospitals, universities, and research labs that we work with. With 141 citations to our name, we're at the forefront of genetic research. Our software, GeneYX Analysis, examines whole genome, exome sequencing, and gene panels. We utilize both short read and long read sequencing technologies, all with the latest ACMG, AMP, and CleanGen guidelines are updated into the GeneYX Analysis pipeline, keeping our software always up to date. With GeneYX, we automate the diagnostic process from sequencer to report using our AI-based genetic analysis solution. So, what can you expect from this course? This course is designed for professionals like yourselves in the genomics industry to advance your knowledge and provide clear, practical experience and examples. In this course, you will have the opportunity to learn from two distinguished experts in the field of genomics, Dr. Hagar Moshaked and Dr. Ali Tabish. Hagar brings a wealth of experience as a senior variant scientist at Hadassah University Medical Center, along with her role as Chief Product Officer at GeneYX. And with her expertise, she has played a pivotal role in advancing genomic research, and this has translated into helping us grow GeneYX analysis platform into what it is today. Ali is another expert joining us, serving as a senior variant scientist and clinical geneticist. With a passion for genomics and personalized medicine, Ali specializes in clinical evaluations of patients and families with suspected or known genetic disorders. His expertise extends to next-generation sequencing data analysis, bioinformatics, genotype, phenotype correlations, variant curation, clinical reporting, and genetic counseling, all in accordance with the latest of the ACMG AMP guidelines. So through this course, you will gain insights into the latest advancements in genomics, personalized medicine, and crucial role they play in patient care. You will also learn practical skills in NGS data analysis, variant interpretation, and clinical reporting, equipping you with essential tools you'll need to navigate the complexities of genetic disorders and provide personalized care to your patients and their families. So here, what can you expect? The GeneYX course runs from the 5th of March to the last Tuesday of April, which is the 30th. We highly recommend that you do not miss any lectures, as each lecture builds on the knowledge and skills you need. Please ensure your microphone is muted throughout all the sessions, and feel free to use that chat feature to ask questions and share insights during the presentation. I will send you a recording of this lecture in the next few days. We have decided to pre-record our lectures as we have a large audience, but we will always hold a live question and answer session after each lecture with both Hagar and Ali. So I hope this has given you all the information that you need. So without ado, if there's anything you need, please don't hesitate to reach out to me if you have any questions. And it's over to Hagar for the first lesson.
So hi, everyone. As Jade said, we have pre-recorded a session, so we are going to hear it now. It was just recently recorded, and I'm going to be here.
Wait,
enjoy. No, go ahead.
Sorry, Hagar. Go ahead.
Yeah, you can start.
Okay.
Jade, thank you for the introduction. I'm very excited to be here. I'm Dr. Hagar Moshaked, and I will start this course of clinical variant interpretation. So we will start with a few slides of introduction, and we'll start with the rare diseases, because the main focus of this course would be how do we diagnose patients with rare diseases? So rare diseases are actually more common than we think. They are defined as those affecting a small percentage of a population, fewer than 200,000 in the US, or fewer than 1 in 2,000 in Europe. We have more than 7,000 diseases that are classified as rare, and this is growing rapidly as we discover novel diseases every day. And although rare diseases are rare, they're actually much more common, because if we combine them all together, we have more than 300 million people worldwide affected with a rare disease. So jumping right into variants type, in which we want to understand what type of genetic information we are looking on and could cause a disease. There are multiple types of genetic variants, but altogether they are categorized into two. Small variants, which are less than 50 base pairs in length, this would be referred as SNVs, single nucleotide variants. And there are large variants, which are larger than 50 base pairs. These would be referred as copy number variants, abbreviated as CNV, structural variations, abbreviated as SV, and repeat expansions. Now, these abbreviations are very important, and we're going to use them frequently. If we look here on the table on the right, again, the variant category, small and large, and then we have the different types of genetic variations. Now, this is the basics of genetic analysis and genetic variants, and I encourage you to all to learn and to know these deeply. Now, we are going to focus on the first few lectures on small variants, and then after that, we would also touch some large variants introduced by Dr. Ali Tabish. So another slide of introduction would be looking on the basic gene structure. The most genes in the human genome are composed with the same structure. 5' UTR, which is untranslated region, upstream to the gene. And then we have exons in our orange boxes here, and then exon, intron, exon, etc. And this could be a single exon gene, could be many exons with even dozens of exons in a single gene. And then there is the 3' UTR, which is again untranslated region found downstream to the gene variant. Now, these terms are very important, and for those of you who are not familiar or not coming from the field, I encourage you to learn these. We describe variant effects, meaning the effect of a specific variant on the human genome in these terms, and we segregate them into coding and non-coding variants. Now, if we look on the coding region here in the exon, we can see here this box with different effects type. And just as an example, there are synonymous variants which do not change the amino acid, and then there is a missense variants which change the amino acid in the protein. We also have types of effects that are predicted to cause loss of function, which are stop gained when a stop nucleotide or a stop codon is inserted in the middle of the gene. And then there is frame shift, in which the open reading frame is being changed. And there are splice donor and splice acceptor, which would cause or predicted to cause abnormal splicing. Now, these 4 effect types, splice donor, splice acceptor, stop gained, and frame shift variant, are considered to be loss of function variants. The splicing variants or the donor and acceptor variants would be on the junction between the intron and the exon. Other types of variants or of effects of variants are in non-coding regions. These could be in the upstream region, in the downstream region, or what we call deep intronic or spliced region variants, intron variants. And there are different types of effects, and I encourage you to learn these schemes. It's a great diagram to present all types of effects that we are familiar with. Now, this story actually begins in the genetic counseling. So I'm taking a step back, and I want to take us to the session of genetic counseling for a slide. Now, the genetic counselor would take a pedigree of the patient, and the patient could be the person itself, or it could be a child, an affected child with the parents. And as part of the genetic counseling is to take a pedigree and a medical history of the specific family. I will take it into granted that all of you are familiar with the pedigree and know how to read it, but briefly, we provide a circle to a female and a square to a male, and the shape could be colored by different colors or patterns to describe a specific disease or a phenotype. Now, during the genetic counseling, the genetic counselor eventually would refer the patient or the parents to a genetic test. And while doing that, there are a few considerations. The first one is the suspected disorder. For example, if this is a Down syndrome patient, usually it's quite obvious, it's clear by the dysmorphism of the patient, the way that the patient looks. And then for these specific phenotype, probably the genetic counselor would refer the patient to perform a karyotype. But in most cases where the genetic disorder or suspected genetic disorder is not that clear or obvious, and we want to provide a diagnosis to this patient, then the genetic counselor would refer to a broader test or a more advanced test. Now, when doing that, there is a consideration of the costs. Obviously, there are different costs and there are different funding in different countries. So in some countries, the NGS analysis, the most advanced genetic test, would be funded by the government. And in some countries, this would be out of pocket. And of course, this is another consideration. And lastly, and most importantly, of course, is what is the available tests to the lab? Where can I send the sample to? Not every genetic test is available worldwide, obviously. In most countries, I would say the advanced countries, the first tier tests for every patient that is predicted or suspected to be affected by genetic disorder would be CMA. This is abbreviation of chromosomal microarray or CGH array. This is a great method to analyze or to detect large copy number variations in the human genome. And then there are the exome sequencing or genome sequencing, which are the NGS genetic tests. And these are the most advanced genetic tests that we can offer a patient these days. And this is the focus of this course. The sequencing or the NGS analysis is divided into two. The wet part, which is how we generate the data. How do we sequence the DNA molecules that are extracted from a specific patient? But of course, there is the dry part, which would be the analysis, which is the focus of this lecture. So I'm not going to provide details on the wet part, sequencing by synthesis, but and this is just one method of sequencing, and we know that there are advanced technologies to sequence molecules that is different from the what Illumina is doing, which is the most common technology used worldwide. But there is a video here, a great video of the basics of NGS sequencing, and I really encourage you to watch this video as a complementary data for this course. I do want to focus on a comparison between exome sequencing to genome sequencing, and this is very important and fundamental to this course. So in exome sequencing, we would sequence only in all the coding regions. There are about, and this is really differs from one genomic build to another, but there are about 180K exons in the human genome, which is about 1.4% of the genome. And if we look on the length of these regions altogether, if we combine them all together, this would be about 30 megabases in length. Now, exome sequencing increased the throughput capabilities because we can sequence many patients in one flow cell, for example. And it's in terms of effects versus cost, this is an optimized approach per sample because we sequence only 1.4 of the human genome and not the entire human genome. So we focus only on the important, what we think to be important regions, which are coding regions. And this is also maximized data storage because if we perform whole genome sequencing analysis, we have to store a lot of information that in most places we don't really know how to address and to analyze. We don't analyze the entire human genome. So in exome sequencing, we would analyze the entire material or data that is being produced by the method, by the test. And this is a great illustration to show that the exome is about 1% of the entire human genome, and again, composed only of the coding regions. Now, when we're doing genome sequencing, this would also include the non-coding DNA that could be introns, promoters, enhancers, centromeres, telomeres, and many regions that previously we used to refer them as junk DNA. We now know these are not junk DNA. These are probably regulations regions that are important for the DNA and for the chromosomes to function. Now, the sequencing technology of the genome sequencing allow better calling of complex variants. And this is very important because when we are doing an exome sequencing, this is a PCR approach, which means we need to amplify the coding regions before we can sequence them. But when we do a genome sequencing, we need less throughput. We need less material to begin with. We need less DNA. And we do not need to perform a PCR, and this just allows better calling of complex variants because we don't have the PCR biases that we all know. The genome analysis would also allow to discover novel genomic variants like structural, single-nucleotide, insertion, deletion, copy number to a better extent. So if complex structural variants are quite limited to detect by exome sequencing, with genome sequencing, after a negative exome, there is a chance to discover variants that are invisible in the exome sequencing. One example to this would be an inversion of the DNA. So if we have a sequence that was inverted, this would be completely balanced. In the exome sequencing, we would not see anything. But in the genome sequencing, we would be able to detect the boundaries of the inversions quite precisely. Of course, dependent where are these breakpoints, if they are in a region with high quality sequencing capability or low quality. Now, all of these considerations of QC, quality of sequencing, are part of NGS analysis and are very important to learn. Now, I do want to take one step back because sometimes there is a confusion between de novo assembly and resequencing. In this course, what we are focusing on, and when we are trying to diagnose a patient that comes into the clinic, we are doing resequencing, which means we are taking the genomic DNA, we shear it, we sequence it, and then we align it to a reference genome that someone already sequenced before. Now, the first time that you want to generate a reference genome, you have to do a de novo assembly. Now, the de novo assembly is to be able to construct the genome sequence from scratch, from de novo. And every time we see a novel creature that we haven't met before, we can take the genomic DNA, sequence it deeply. So this will require high coverage. And then we would try to align the pieces, to connect the different pieces of DNA molecules to create one continuous DNA molecule that would represent the genome of this creature. Now, this is being constructed by the overlap between reads. In comparison to that, resequencing would mean to align the reads that are being obtained from the patient, from the individual that is being sequenced, to a reference genome. So if we look here on the gray bar, this is the reference genome. The yellow reads are here. Every line here is a molecule of DNA that we sequence. We call it a read. And the coverage is the entire reads on a specific region. Now we try to align it in resequencing, we try to align these reads, these molecules of DNA to a reference genome. And then using that, we try to identify variants, genomic variants that are not found in the reference. So these would be deferred, and we would call it alternative versus reference. Now, these terms are very important for every genetic variant that we would call during the analysis of an NGS. We would find the chromosome position, the reference variants at this position, and the alternative variants of your patient at the same position. Now, during time, the human genome was sequenced, de novo sequenced several times, and we refer to these versions as genomic build. Now, 2010, the AG19 was released with a few updates after. 2015, AG38 was released, and this is the most, I would say, commonly used. Both versions are used, but 38 would be highly used in different labs today. And lately, in 2020, the latest version of the human genome was released. This was called T2T, telomere to telomere. There are great publications that I encourage you to read how we improved the de novo assembly technology using through time. I would just hint to say that the technology of sequencing was improved by enabling long-read sequencing, which means that now I don't have these smaller pieces, but I have a long, continuously DNA molecule that improves dramatically the capability of doing a de novo assembly. So now when we understand the difference between de novo assembly and resequencing, we would focus on resequencing in this course. Now, finishing with the introduction, I want to start with the bioinformatic analysis. This could be some information that some of you are less familiar with, and we'll start here. So let's say I sequence a specific patient. This goes through the wet part, the actual sequencing, and then we put the sequencing in the sequencing machine, and the machine would issue a file, an output, the first output file of the sequencing, the raw data file. If we talk specifically on Illumina, Illumina technology would be BCL, also MGI technology. Now, BCL is the first output from the sequencing machine, and this would be the first process, the bioinformatic process, we call it primary pipeline, would be base calling. And the BCL contains some visual information from the sequencing itself. Now, the process of base calling would be sequencing signals is transferred into nucleotides. So from fluorescence information, from visual signal information, we would convert that into nucleotides. And this would result, or we call it BCL to FASTQ. So FASTQ would be the output of the primary pipeline. Now, this slide focuses on the main technology in the market, which is Illumina, but different sequencing technologies like PacBio and Oxford Nanopore for long reads have different types of primary pipeline, which means different types of raw data file and the output. So, for example, for Oxford Nanopore and PacBio, instead of a BCL, we would have POD5 files. So this figure or this slide is specific for Illumina. Now, starting with a FASTQ file, now we can move ahead to the secondary pipeline. And the next process, the bioinformatic analysis that we would do would be an alignment. The FASTQ file contains many reads of DNA, which is just the sequencing of the entire reads that were sequenced for a specific individual. Now, the process of alignment would be aligning these reads, to a reference genome. We just describe it as a resequencing or reassembly. Now, while we do the alignment, we produce a file that we call BEM file. And the BEM file is also a visualization file. It enables me to visualize the reads of the DNA by IGV or other visualization tools. So alignment would be converting the FASTQ to a BEM file, and this is the secondary pipeline. The next step would be a variant calling. In the FASTQ file, we have the entire stretches of DNA molecules that were sequenced by the machine. But obviously, I do not care about the entire raw data file. I'm actually looking for the regions or the variants, the positions in which there was a difference between the alternative and the reference. Now, this process to call for the variance, we call it a variant calling process. In this process, we extract differences between samples to the reference genome. So every position in which the reference and the alternative is not the same, we would call a variant and put it in a file that we call VCF, that is called VCF. A VCF is a variant calling format as a list of variants. So while in the FASTQ files we have a list of DNA molecules with the entire sequence of these reads, in the VCF we have a list of variations with chromosome, position, reference, alternative, and other QC information in the format field. The VCF file is a standard file. All secondary pipelines should issue the same type of a VCF file. It's quite standard, which was a great advancement in this field. So secondary pipeline, now starting from FASTQ, we produce a BEM file, or it produces a BEM file per visualization of the reads and a VCF, which lists all the variants in the specific sample. We also have QC data to present or to describe how well was the sequencing, what is the average coverage, what was the mean coverage, what is the, so many of the parameters that we use for QC would be, what is the percentage of the exome that was sequenced above 20x, about 50x, about 90x, about 5x. So these parameters would be used to describe, and these are also standard, to describe the quality of the sequencing of the specific sample. And of course, there are some thresholds for QC data that are considered to be high quality and low quality. So just as a thumb rule, for exome sequencing, average coverage of accepted of high quality data would be about 80x or 100x, depends. And for genome sequencing, this would be 30x, so much lower. And this goes to the differences between exome and genome, as the exome is composed of a PCR, of amplification, and genome is not, and is much more unified across all the human genome. Now, the next step of the secondary pipeline would be annotation. The annotation could be considered to be part of actually the tertiary analysis. It really depends. But the process is that you take every variant in the VCF file and you annotate it. Now, to annotate means to provide data on a specific variant from different sources. The annotation of the variants is being done using different databases and tools, and the result of this would be an annotated VCF. Now, at this point, you would have a VCF with a lot of information embedded inside. This would again be referred as annotated VCF. For an average exome sequencing of an individual, you would have here about 100 or 200,000 variants. Now, obviously, this would be extremely hard to start manually analyzing this file. And for that reason, there are different types of approaches and different approaches for filtering the annotated VCF. Now, the annotation and the filtering would be considered as the last part of the pipeline, which is the tertiary analysis. And the process here would be to filter the variants based on different criteria. This could be allele frequencies, this could be conservation, this could be QC data for every variant. And the result would be a filtered file, which would contain a shorter list of variants, which at this phase, the analyst comes into the picture, sits in front of the computer and start reading and analyzing the data. So the last part would be VCF to an annotated VCF, and this is where the bioinformatic phase ends and the manual analysis starts. And this would be our focus. Now, when we do NGS analysis, we have different considerations. So let's say I'm sequencing a specific patient. I've ran it through sequencing, I've ran it through the bioinformatic pipeline, and now I have the annotated VCF file in front of me, and I need to analyze it. There are a few high-level considerations that needs to be taken when we begin this. And they are all concentrated around the patient. So first would be, what is the phenotype of the patient? Some phenotypes are well known to be recessive. Some are known to be dominant. Some phenotypes are known not to even be genetic, or some are known to be polygenic, which would make my analysis much harder. All of these considerations are very important. So every time I get a referral letter and the data which are linked together, I ask myself, what do I know about this phenotype? Is this a known phenotype? Have I seen this combination of phenotypes before? And of course, for seniors, or for physicians, this part would be easier. For people that are not coming from a clinical background, this could be harder. But of course, you learn with time and you get to experience different types of phenotypes and learn what is the inheritance model expected for some of the phenotypes. Just as an example, hearing loss could be dominant and recessive, and this is well known that both inheritance models exist for this phenotype. But other phenotypes are well known to be X-linked. For example, if we suspect Fragile X syndrome because of the dysmorphism of the patient, we would immediately go and test an X-linked gene. So the phenotype directs us in what type of genetic test to offer, but then even after we choose an exome sequencing or a genome sequencing, it would direct us on how to analyze the genetic data. So how do we even describe phenotypes for genetic analysis? A standard dictionary for this would be the HPO. HPO is a human phenotype ontology. It's the most standard dictionary worldwide for genetic analysis. It is highly used. So based on my experience, every genetic lab that I've seen before, and there are hundreds of publications on using HPO, would use HPO dictionary to describe phenotypes in the patients. Now, what's very nice about the HPO dictionary is that it is built like a tree. So for we have subterms and we have terms that would describe systems. So for example, we have this, which is phenotypic abnormality, and under phenotypic abnormality, which is the main term that every other term would sit under, we have growth abnormality, for example. And under growth abnormality, we go and we define it, or we further specify the phenotype that we see in the patient. This could be abnormality of body weight, which would be further described as tall stature or short stature. So for every subterm, I can go up the tree to try to understand on what system am I working on. Now, these branches have this overlap, which creates further complexity. So as you can see here, short stature can go under different terms of HPO. This method is very important for a few reasons. The first one is we want to have a standard dictionary, a medical dictionary, and in other departments in the hospital, they would not use HPO. So HPO is very specific for genetics. If one patient travels from one lab to another, the phenotypes that describe this patient should be standardized among all labs. And this is also very important when you want to compare different patients. So if, for example, I could have one term with synonymous with different terms. For example, hearing loss and deafness. These both terms would describe the same phenotype, but when I want to compare different patients, I have to know which terms am I using, because I'm using here so machine learning methods and using automated comparison, and I have to know the synonymous of all these terms. So using HPO is highly recommended wherever lab you are at, and I would highly encourage you to visit their website. They have great tool that they call Phenotype, the Phenomenizer, in which you can put the description of your patient, and then the tool would provide you the related HPO terms that you need to use. So this is how we describe the patient using HPO. And then we have genes. So looking on all the data, the NGS data, with the patient in the middle, I want to learn about the genes that the variants that I found is at. So the genes, many of the genes in the human genome were already described to be disease-causing, and for that reason, I could use this information while doing the analysis. So just as an information on gene names or gene symbols, we use AGNC to provide the most updated gene symbol. Genes are changing their names through time, which is quite confusing. For a lot of time, this created a mess because when you want to compare two sequencing technologies or two bioinformatic pipelines, you have to be aware to the fact that genes could be described by different names. We call them synonymous or aliases. So aliases of the same genes currently would be described all together under AGNC. This foundation is responsible to give names to genes. We are still naming genes, not after researchers and physicians that discovered novel diseases, but actually we name them after their conservation or other related proteins, their function in the human genome. And we have many genes that we haven't named yet. These would be usually called as C9, for example, or F7, which mean chromosome number. C9 would be the chromosome number 9, and then ORF, which is open reading frame, and then a number. So whenever you see this expression, and we have a gene that is well known, C9orf72, that is causing ALS. This is a very known gene, but the structure of these genes means that we haven't named these genes yet. If you want to discover or learn more about gene names, you can go here to HGNC. Next, we look on the variants themselves in the list of the annotated VCF. Now, we also have a standard way of describing variants, and this would be the HGVS nomenclature. This is an internationally recognized standard for description of DNA, RNA, and protein sequence variants. It is used to convey variants in clinical reports and to share variants in publications and databases. So we all understand we need to standardize the way that we classify variants, the way that we describe variants, and the way that we call the genes that these variants are found in. So the AGVS has a very clear guidance here in this website. How do we need to name a specific variant? And for example, here I put a screenshot from the GeneYX platform of a specific location, a specific gene, a reference alternative amino acid, and the AGVS nomenclature. Now, so far we just described the variant calling format and the annotated one, but we described the location, which means the chromosome in which this variant is found in the specific position in the human chromosome. Now, please note that this is genomic build specific. When I describe a variant, I must say in what genomic build am I describing it? Is it 19? Is it 38? Because the location would be different, and I cannot compare variants that come from 19 straightly to 38, I would have to lift over or lift under these variants to convert them from one genomic build to another. Now, I also have the gene name. This is the TYR gene, the tyrosinase gene. It is a very known gene because it causes albinism. And then I have the reference variants. So at this specific position in the genome build, in the reference genome build, there was a C, a nucleotide of C. But in my patient, I have an A. And for that reason, this is called as an alternative. And then there is the amino acid change. But the correct description of this would be the AGVS, as I've just described. AGVS would be C dot to represent the variant on the mRNA level. And then there is P dot, which would be on the protein level. So C dot 575 C2A would mean that when we start from the first codon, the start codon, and we start counting the nucleotides, at nucleotide 575 on the mRNA, there was supposed to be a C, and now there is an E. And then on the protein level, there was a serine at position, of the amino acid position 192, which was converted to tyrosine. Now, this conversion of amino acid, if we described effects at the beginning, this is a missense variant because it changes the amino acid. So we always describe variants using the AGVS nomenclature. This is what we put in the clinical reports, and this is how we share variants among different researchers, publications, and databases. So as part of this first class or first lecture, I did wanted to provide some high-level information for some of you that are not straightly or directly coming from the field. But NGS analysis types, there are many different types or reasons why we want to do NGS analysis. So we started by describing a patient that is going to a genetic counseling session. But sometimes it's not, we are offering or we offer genetic tests not due to a specific disorder in the patient that comes into the clinic. But this could be a carrier screening. This could be because of a family history of cancer, for example. Now, to summarize this, I've provided an overview of different genetic tests that are being offered in labs today. This, of course, is different from one lab to another. Some genetic labs would offer all of those tests, some would offer many more, and some would offer only a single genetic test. But if we review it really fast, we would see that these are divided into germline, panel, somatic analysis for somatic variants, and then maybe health screening tests. Now, under the germline tests, there are exome sequencing, there are genome sequencing. This could be in the framework of a single patient being sequenced or a trio, which refers to a mother, father, and the proband. So when I have the entire trio, I am able to analyze the data in a much better way because I can compare the specific patients to the mother and the father. Another types of genetic analysis that we offer are carrier screening. This could be couple screening. This could be a single or a solo carrier screening for an individual. We also have the mitochondria analysis, germline analysis for structural variations, for example. And we have long read sequencing. This could be Oxford Nanopore or PacBio. Panel design or under panel, we have different gene panels, and there are hundreds gene panels that are available. One of the very nice tools to design a panel of genes would be the panel app, and I encourage you to go farther and look for this panel app. We'll put the link in the chat. And gene panels can be virtual gene panels, which mean we would sequence the exome sequencing, but then we would analyze a set of genes and not the entire exome. So we would filter out everything that is not on our closed list of genes, And there are panels for autism, panels for neurodevelopmental disorders. There are panels for hearing loss, for visual issues, etc. So there are hundreds types of panels. And basically, every lab could define the lists of genes in every panel. There is no standard panels around, and there is no one panel that everyone is using. Every lab would offer the list of genes that are part of the panel, usually in their websites. We also have cancer panel, which is just one type of a specific panel. And there are, if we're going to the somatic region, because we started with cancer, we can also sequence the tumor. We can sequence, so cancer panel would be to detect inherited predisposition to cancer. But somatic analysis would be to affected individuals with tumors, with cancer. And for these patients, we can offer tumor sequencing. So we would sample the tumor, and we would sample the near normal tissue, and we would compare tumor versus normal. We can also just sequence the tumor itself, and so SAM labs would sequence it through time to track the processes of the cancer, the clones that are being generated or disappeared through the use or prescription of specific drugs and treatments, or the remission of the disease. And there are all the health screening gene tests. I only have one here as an example, which is the pharmacogenomics, PGX, abbreviated as PGX. PGX offers the detection of drug sensitivities or adverse effect to specific drugs. So we all know that certain genetic variations cause inability to metabolize specific drugs or have a slower metabolism for a specific drug. And this all goes under the pharmacogenomics, which in the last few years is a great hit. Under health screening, there are also some well-being tests. These could be nutrition tests. As a scientist myself, we only do some scientific work here, but of course there are hundreds of genetic labs focusing on well-being, on detection of relatives, and there are endless number of companies focusing in this region. But as a geneticist, I will always encourage you to test what scientific knowledge and data do you have behind these tests before you go and take them or you perform them. Some of them are highly validated, some of them are less validated. When we do a genetic analysis, we can also discuss for one slide the clinical versus research genetic analysis. So in genetic labs that are found in hospitals or clinical labs, we would perform clinical tests. But in academic institutions and also in many hospitals that are affiliated with an academic institution, there are a lot of genetic analysis that are being conducted under research and not a clinical test. So I think it is very important always to know, what are you performing? Is it a clinical test or a research test? Because there are clear distinguishments between the two approaches to sequence a specific patients. So for clinical tests, the purpose of this would be diagnostic, would be to provide disease risk assessment, for example, in carrier screening, and maybe to personalize medicine using pharmacogenomic analysis. Now, this would be diagnostic. The regulatory would be, you would need a certification to offer a clinical NGS test, and you would need to perform sometimes some validation studies. That isn't compared to research NGS analysis, which usually the purpose is scientific discovery, for example, detecting novel genes, novel diseases. This could also be population genetics. For example, if we want to sequence high volume of healthy individuals to serve as a background to detect so we could combine the allele frequencies for different ethnical backgrounds. This would be a research effort, not a clinical one, and to perform some functional genomics. All of this would go under the research NGS analysis. And when you do research NGS analysis, in terms of the regulatory oversight, this would be ethical guidelines. So you are bounded into some restrictions that are ethical, and you would need to have an IRB approval versus a certification that is different between one country to another. So most of the hospitals and genetic labs are affiliated to a specific IRB committee, and they have to submit and approve these research efforts. In terms of data analysis for the clinical purposes, this would be standardized protocols. You have to be robust. You have to use the same protocol for all your patients, and you have to have a validated pipelines. So you cannot use a random pipeline, and you cannot change it from one patient to another, because there is always the risk of missing a diagnosis for a patient. Under research, this would be more liberated. You can have a custom analysis pipeline. You can develop your own algorithms. This would be commonly used. Every lab will have the in-house algorithms. We call them home brews. And this is highly used for research purposes. This is less recommended when you want to have a clinical lab in which you do need to have certifications and a standard protocol, and of course, a validated process. In terms of the interpretation, so we have for clinical NGS, there is a clinical significance. We are not just sequencing and analyzing, diagnosing patients because we want. There is a clear purpose for this. We will provide them clinical insights. For example, what is the best treatment? What is recommended? What is the prognosis of the patient? There is a patient-specific context into the interpretation. For example, if this is a live pregnancy and we test the fetus, there are clear implications of the genetic analysis that is being conducted. Under the research NGS analysis, this would go hypothesis generation or hypothesis-driven, maybe to learn biological mechanisms. Usually, the interpretation could take a lot of time. This is not something with clear implications. There would not be clinical implications to a research analysis when it is research. In terms of reporting, so clinical NGS would issue a structured report for healthcare providers, for the patients themselves, while the scientific analysis or the research analysis could end with a publication or a conference or a presentation. And this is maybe the moment to say that most of the diseases that we discover, we start with research, we publish them, we present them into conferences, and only after they are validated and published, only at this point, we will convert them into being a clinical test or a clinical information, and they could be reported under clinical settings. Now, lastly, the communications. When we talk about clinical NGS, there is a clear healthcare professional. This could be a genetic counselor, it could be a geneticist, but we are all concentrated around the patient care decisions. The outcome of the clinical analysis would be delivered, will be reported to the patient. And this is not always the case for research NGS analysis, because mostly the communication would be towards scientific community, collaborations with other researchers. And the patients, most of the time, would be reported or disclosed with the information only after this information would be converted from research to clinical. Now, I hope that I made a bit clear the distinguish between clinical NGS and research NGS. In our course and this effort here, we are mainly focusing on clinical NGS analysis, but of course, all the tools that we will learn, all the databases serve for research and clinical analysis all together. Now, before the end of this lecture, I want to show you some use case or a case report of a nice story, because as I said, we start research, we go clinical, and in most clinical labs, there is a research effort being conducted. So this story begins with a child. This child, we see here the pedigree of this family, and we see here marked in black the specific patient. This patient had a developmental delay in autism. It was a male patient, we can see here the square, and this patient was born to a consanguineous family. We see here a double line, a double cross here would mean that the two parents are related, consanguineous. Now, the family history of this patient included an affected offspring, another fetus of this patient that presented AV canal, an atrioventricular canal, suspected with absence of the thymus, and also intrauterine growth restrictions. We call this IUGR. Now, this combination of presentations in a fetus have led to actually the abortion of this fetus. But the family history was significant, and overall there was a quad exome sequencing that was performed. The affected patient, the fetus, because there was a sample, a DNA sample from this fetus, and the two parents. Now, while doing a clinical analysis, we couldn't find anything. So when we can't find anything on the clinical analysis, we would convert into a research setting in which it will take more time to analyze the case. It's not a robust analysis, and we would try to use different tools and databases. In the research analysis process, we discovered a homozygous loss of function variant that was found in a gene named WBP4. Now, WBP4 is a spliceosome-associated protein, which means it is part of the spliceosome. It is responsible for splicing, accurate splicing of genes, but it was not described previously to be disease-causing in human. Although it wasn't described to be disease-causing, it was highly suspected as it was homozygous loss of function variant. We also refer to homozygous loss of function variant as knockout. So this is actually a human knockout, this patient, to the WBP4. And of course, this is all predicted because we only see the effect on the variant level. We do not know the actual outcome or the function of this variant. Now, at this position, after we performed segregation in the family, it was clear that the parents are heterozygous and the affected individual does not have any functioning copy of the gene. We put the gene in GeneMatcher, and it revealed overall 8 families. So GeneMatcher is a great tool to cross genes from one lab to another. It's a matcher of genes, so if you haven't heard of it, I encourage you to go online, Google GeneMatcher, and try to find it and create your own login, because every time you suspect a gene, but you have a significant consideration for this suspicious, so not every gene that you see, but if you're working on a gene as a researcher, if you have a family and you suspect a novel gene, you may put it into GeneMatcher to find if other researchers, if other clinicians worldwide are suspecting the same gene. And then after putting the gene, you would receive an email with the hits. That's how they call it, hits. And then you may address these researchers to discover more information. So with that method, we revealed overall 8 families, which was very exciting to us. So if this is our family, in the family that I was responsible for F1, we discovered overall 8 families, and they come from different regions in the world. Interestingly, 4 of the families had the exact same variant, which was a deletion of an A at nucleotide position 499 in the mRNA. Now, deletion of an A is a frame shift because it's not divided into 3, and that is expected to cause loss of function. Other families had different variants, but they were all loss of function variants. If we look on the WP4 gene scheme, we would see, we saw it at the beginning of this lecture, the 5' UTR, the 3' UTR, the different exons, and we see that all the patients had loss of function variants at around the same region or domain of the protein. Now, this was a great success. It was published, this paper, of course, with an additional function or mechanism that was discovered for this gene. And we discovered that the gene is affecting, or the loss of function of this gene creates abnormal splicing because this is a spliceosome protein. And the phenotypes of these patients was very challenging because it differed from one patient to another. But all patients showed motor delay, speech delay, and hypotonia. 6 out of 7 that we had the information about they had abnormality of the outer here. We can see it here, the dysmorphism in the pictures, the photos of these patients. And we also had intellectual disability on all the available to test patients. So while there were many features that were patient-specific, there were also some common phenotypes. Now, this work was published recently. You're welcome to have a further read. We called it biallelic loss of function variants in WBP4, and encoding a spliceosome protein results in a variable neurodevelopmental syndrome. We can see here the graphical abstract to just show or present that this loss of function caused altered splicing of the phenotypes associated genes. So we did RNA-seq analysis, and if, of course, everything is in the paper, and you're most welcome to go and have a further read. Overall, the abnormal splicing in these patients caused a novel neurodevelopmental syndrome with variable manifestations in different patients. So with that, I would like to finish the first lecture and give you some time for questions.
Thank you so much, Hagar. That was a really fascinating lecture. I personally especially liked the use case at the end. I thought that it tied everything together really nicely and gave a great example of how you actually apply the information. So I'd love to hear from you. I know that we've got quite a few questions in the chat box. Dr. Hagar, if you'd like to.
Yeah, sure. So first of all, thank you for all the questions and the great discussion on the chat. I took a few questions, and me and Dr. Ali would try to address them. I would start with a few. So someone asked about how do we identify sequence or technical artifacts and false positives in the annotated VCF file. And he also comments on the fact that it was suggested to use IGV, but not sure what are the parameters that we need to look at. And I think this is a great question, because part of the problem of this field, or the challenges that we are facing, is the fact that we not always know to distinguish between a false positive from a real call. Now, there are a few thumb rules for that. First is a QC parameters for the specific variants. So we have QC for the entire sequencing. It could be an exome or a genome, but we always have a QC for every variant that we call, also a score, and a few parameters that comes from the VCF during the variant calling process. Now, there are some thresholds to treat a variant with high quality. It depends specifically which technology and which pipeline you're using, but I would suggest to review the thresholds that are recommended for a real call. The second would be the allele frequencies. We know that after you sequence 100, 200 patients, and you have a significant cohort, the repeated variants that are very common could be either polymorphisms that are common in the specific cohort, but they could also be just an artifacts. And if you see that too many times, then for sure you can filter them out. For copy number variants and structural variations, the rate of false positives is very high, much higher than for single nucleotide variations, as pipelines are still improving. We would address that in the copy number variants lecture, so I'm going to hold with this. But just a comment on the IGV. IGV is a great tool to look on the variants and to try to assess whether they are real or not. We use that all the time. If you start from a VCF file and not from a FASTQ, you must be able to look on the BEM file using IGV or a different visualization software. In IGV, low quality reads would be marked. We would see them during the next lectures. So we would definitely address this issue of how do I know whether in IGV I see a real call or not. So thank you for the question. Now, the next question was about the coverage. It was how much coverage is enough to analyze germline and somatic samples. And I want to address these questions to Dr. Ali. Can you share your thoughts on this?
You've got to take off your mute. It's always the mute. There you go.
Can you hear me now?
Yes.
Oh, okay, good, great. Thanks, Agar. So the question is, what should be the coverage for whole exome or whole genome sequencing analysis for the NGS testing? So it depends. If we are doing a germline analysis like Mendelian disorders, it's highly recommended it should be above 20x. It's usually above 30x, between around 50x, so 50 times sequenced. And if it's a cancer samples, we know cancer samples have a different tumor cells. There's a tumor heterogeneity. So we have to take that factor into the account. And for cancer cells, the coverage is usually much higher than Mendelian. It's usually by a factor of 10. So if we are doing a 50x for Mendelian disorders, we usually sequence them for 500x for the cancer samples from the somatic tissue. I hope this would answer the question.
Yeah, so just to elaborate on this, for a genome, it's usually the average coverage would be the 30x. For exome, it would be 100x. And for a somatic panel of a shorter list of panels, usually that would be around 500x, as Dr. Ali was saying. And now there is a follow-up question: what should be the minimum recommended variant depth for reporting? And I think this really differs from one lab to another. I can say that in my own lab, we only report above 10x. So 10 reads, 10 coverage of reads in the same position for a single variant before we report it. Some labs would go with 8x. So this is another number that we heard of, but I don't think there are clear guidelines of how you should handle or what is the number of repeats or reads, but it is also different between a genome and an exome. So the numbers 10x and 8x usually go for exome sequencing per genome, as it's more unified and the coverage is lower from the beginning, then you can go even lower. Okay, there are a lot of other questions. Let me just bring another one. So someone asked about a recommendation for a lecture to have a better understanding of HPO and the phenotyping in HPO. I would say we would definitely address that in the following lectures. The next one, I would describe the Phenomizer, which is a great tool to try to translate a clinical presentation of your patients to an HPO term. So we would address it more deeply. And there are many publications and probably online lectures about this. Another question, I was referring to the research that I've showed you at the end, the case report, and that was about the transition from clinical to research, exploring variant of unknown significant variant. So once we have a variant of unknown significance, but it's positioned in a novel gene, which means a gene that was not described before as disease-causing, we would not call it a VUS, but we would call it a GUS, gene of unknown significance. Now, GUS variants, and this is not highly used, it's kind of our terminology, I guess, but GUS variants would be shifted from clinical to research standards. So this is when we switch. In terms of, so the follow-up questions on that was, do we simply expand panels to exome or genome? So in my own lab, when we offer a virtual panel that is based on an exome, we would, if it's a negative one, we would expand or unfilter the specific genes for the panel, and we would look broader, and it still could be clinical. So this is not what distinguishes a clinical from a research test. I see that someone said he would adopt the goose terminology. Yeah, go ahead. How analysis differ from standard GATK? Ali, do you want to take this, or should I? I will just say what we have in GeneYX, and then maybe you can elaborate. So GATK, most secondary pipelines are based on GATK tools, so it's kind of the same. But in GeneYX, we have Dragon and Sentient implemented. Both are great secondary pipelines. Dragon is the official pipeline of Illumina company, and this is a highly used with high standards and validation process. So we use Dragon. It is considered better from the common publicly available GATK. It's very expensive, so this is not a free of charge like GATK pipelines. They have a great copy number variations and structural variations, and for this, which greatly improves with the Dragon pipeline, I would use that. Okay. Okay, so one of the questions now in the chat is best practice in gene panel design. I've posted here on the chat a link to Panelab. Panelab is a great foundation that curates genes for panels. You can search a specific panel, specific phenotypes, and find a curated list of genes for the specific panel. For those of you who know Panelab, I think maybe don't know that there are two Panelabs. There are UK and there are Australia. Now, I've just heard recently, and this is not highly based, so don't take it for granted, but we heard that the Australian one is much better because it updates more frequently. Now, when you go online, and I've posted you the URL of the Australian Panelab, you would see the log file, and they update every day novel genes for every panel. So yes, this is a great way to construct gene panels. Another way would go online and search for publications, but this would be a long way when you want to define a specific sets of genes for, yes, I see again the link, thank you. How can we report a variant under 20x? The confidence would be low for the variant. What would be your suggestion? So I think a variance that is under than 20x could be with high confidence score. It really depends if it's a genome. So 20x would be perfectly fine. If it's an exome, it could be low. But usually it really depends if we know the variants before. So if I know the variants and it's a founder variance, for example, and it's a region with low complexity, harder to sequence, and we will talk more about this while the course goes on, because there are regions where it's much harder to sequence than other regions. And then 20x would be perfectly fine because I know the variants, I've seen it before, it fits the clinical presentation, but I would most definitely recommend a validation. So I think in the past, 2 years ago, we used to validate every variant that we reported from exome or genome sequencing because the technology was quite new in the clinical setting. Today, if we have high quality, we don't even recommend validation. Okay, so there are many other questions. Let me just try to grab more of these. So one really interesting question was, what is the difference between coverage and depth? And actually, I got to think for a second, but coverage usually refers to the entire human genome coverage, how the entire test was covered. We usually describe the average coverage and the mean average for exome sequencing, for genome sequencing inside the report. And the depth would be by variant. So what is the depth of a specific variant and not the entire region or the entire sequencing data? So that would be the difference. I see many questions. Let me try to grab another one. Okay, so is there any guidelines on how to analyze long reads for variant classification? So first, thank you for the question, because some of you may know that GeneYX is the official company that analyzes PacBio and Oxford Nanopore long read sequencing data. So both companies have chosen GeneYX to be their official tertiary analysis platform. For long reads data, we start from a VCF file and not from a FASTQ. We don't run their secondary pipeline, which is customized specifically for their data. But so the question about specific guidelines, I am not aware of a specific guidelines for long reads. And now I see a question on the difference between long reads and short reads. So long reads are long molecules of DNA. If in the common method today, the short reads by Illumina, we sequence 150 base pairs to 300 base pairs of a single DNA molecule at one time. So every read of DNA would be contained of 150 base pairs. For long read sequencing, we get to the average number of 15K base pairs. So 15,000 molecules of letters of DNA nucleotides in one read. So long reads is a novel technology. It's not that novel, but more commonly used today. There are two sequencing technologies in the market, PacBio and Oxford Nanopore. Both of the company use different technology to sequence long reads, completely different. If this is, we have a focus on one of the next lectures on long reads, and we will describe the technology a bit better. As I said, I'm not familiar with guidelines that are specific for reporting of long reads data. We are still at the phase that we are trying to analyze the long reads, trying to gather a database to remove all the false positives, variants that are there, because every technology brings its own noise, its own artifacts. So we are still at this phase and there aren't any specific guidelines for long reads. Pseudogenes. So I'm not sure I understand the questions, but oh, you will see us at the ACMG in Toronto, at Canada. So there is a questions about this. That posted from our side. So all of you are welcome, whoever is joining the ACMG conference in March, and then the ESCG at Berlin. We will be there, so we encourage you to come and say hi. In terms of the pseudogenes, so the pseudogenes obviously are duplicated, unfunctioning copies of a single gene of genes in the human genome. And long-read sequencing dramatically improves our ability to differ or distinguish between the real gene, the functioning copy of the gene, and the pseudogene. Pseudogenes were created as part of evolution by segmental duplications. And yeah, I see that someone here is writing that according to the ACMG guidelines, you should report above 8 reads. Interesting whether this is specific for exome or a genome. Maybe it's the same, but 8 reads are, yes, makes sense. So coming back to pseudogenes, long read sequencing improves dramatically the way to align them specifically, and this should be much improved in the next versions of the pipelines of the long read sequencing companies, and we already see great improvements. Also for the pharmacogenomics genes, where you have sets of long structural variations that needs to be called.
Thank you so much, Hagar.
You're welcome.
Have you got some more questions coming up?
No, I see that you wrote yourself Jade, the real one. This is really nice. So there are a lot of Jades here because of the malabuse of the link, but we'll learn from that.
Right.
I guess I just want to say, everyone, thank you for joining, and we're really excited to be here.
Right, correct. And I just wanted to, just to re-emphasize as well that we would really love to offer everybody a certificate after this course. It's a very detailed course, as you know, that we're going to be doing it till the end of